Welcome to Chillin' 101. We hope this video helps.
You begin by locking down the essentials. Know the cell types, molecular basics, and lab safety. Once you have the syllabus and one reliable textbook, map topics to lectures and lab sessions so you study in sequence. Make sure to block 2 3-hour focus sessions with 25/5 Pomodoro intervals. This trains recall and prevents fatigue. Also, prioritize high-yield topics first, like microbial genetics and pathogenic mechanisms, because they recur in exams and labs, so start there and move outward. Now you switch to active learning. Make concept maps connecting taxonomy, physiology, and virulence. Once done, convert key facts into spaced repetition flashcards. Use question and answer prompts that force recall instead of recognition. Make annotated lab diagrams for staining, culture workflow, and PCR cycles so you visualize procedures before bench time. Also, practice past problems and explain concepts aloud. Teaching reveals weak spots and deepens understanding, so spend time on application, not just memorization. Finish by simulating exam conditions, timed practice tests, and mock lab write-ups that mirror grading criteria. Once you complete these, analyze mistakes and create a mini-plan to shore up the top 2 weak topics in the next study session. Make sure to rehearse common lab errors and corrective steps so you reduce repeat mistakes. If things still go wrong, ask your instructor or a peer for a focused demonstration. That quick guidance often clears persistent misunderstandings and saves hours.
Thanks for watching. Consider subscribing if we helped you.